The next topic in general microbiology that we'll be starting with are the microscopes. Now, I've told you a little bit about the microscopes when we started off with the scientists part and who discovered which one, but it's now time to go into the details. So we discuss all of these and then we'll practice a few questions. So going forward, before I start off with all the different types of microscopes that we have, it's very important that I teach you first and foremost the features generally seen for every microscope. So what is the basic purpose of a microscope? That it should be able to, magnify or enlarge or zoom in into something. So every microscope will obviously for magnification use lenses, different types of lenses which I'll eventually come to. But yeah, a lot of lenses have been used. Next, the thing that a microscope should be able to do is resolve. So what is resolve or what is resolution? So for example, if I tell you the exact book definition which you used to read in your school time also, the ability to differentiate 2 points as separate. The ability to differentiate 2 points as separate, that is what is called as a resolution. So obviously the resolution that we have for the human eye versus that the microscope will have, and that too what an electron microscope will have, will be totally different. So for me the resolution is different, and for the microscope it's going to be different. For me the resolution, or for the human eye, it is only 0.2 mm. When I talk about the light microscope, it is 0.2 microns. And when we talk about the electron microscope, it is 0.2 nanometers. That is why have you seen viruses? Viruses are in nanometers. That is why viruses are seen on which microscope? They are seen on electron microscope. Do you ever see viruses on a light microscope? You don't, because that is the resolving ability that it has. So remember 0.2, 0.2, 0.2, but obviously the human eye has 0.2 mm, the light microscope has 0.2 microns, and electron microscope has 0.2 nanometers. After the resolution ability, the next thing that we need to have is a contrast dye. So obviously contrast, what is a contrast? Like for example, you know that the if the background, like you have the background in all your prep ladder videos, so your background is what? Your background is all black. So obviously white color is going to shine out. So that is my basic color combination and common sense, that if there's a black background, then white color, then obviously this white color is going to shine out on top of it, right? So same way I will have to do some color combination. I will have to find out that, okay, if this is a red color organism, let me put the background as maybe blue. If it is a pink color organism, let me put the background as yellow. So that is how I put different colors and I find out the contrast. So simple color theory goes ahead. Repeating that for all of you, every microscope will have a magnification source, it will then have a resolution, and it will have a contrast. Having said that, the first microscope, which is everyone's favorite, which is going to be the mainstay, and now that I have taken this responsibility of teaching you the second year, that is path and micro, so this is the bread and butter for all of us. Okay, microscope is something without which you cannot survive in both of these laboratories, and also something you never switch on when you're in your second year. And I can say that because I'm equally guilty of doing that. The first semester of our second prof, I remember that we were not even aware of how to switch on a microscope or properly focus a slide, and so many times we've broken glass slides while focusing them. So that's a part and parcel of everyone's MBBS curriculum, right? But yeah, having said that, now I know the importance of it, and now I look back and obviously laugh a lot that how from being a person who never knew how to switch on or focus a microscope to a person who's constantly looking into it day in and day out now, that is how destiny plays its role. Having said that, we move on to a light microscope, and their favorite question is labeling of the light microscope. So first and foremost, because you've used the word light, the first question that is going to come to you is, what is the light source? What kind of light are you using over here? We use transmitted light. What we use is going to be transmitted light. What do I mean by that? For example, can you see there's a tiny little light bulb over here? And how is the light going to go? The light is going to go in all the directions, right? The light is going to go in all the directions. So it's getting transmitted, and that is exactly what you need to know out here. So the first labeling that I'll do for all of you, this is the light source. What next? Now, if you noticed, I was saying that light is going in all directions. Light is going in all directions. I don't want that to happen. Why would I want light from a microscope to go everywhere in my room? Not happening. I'm not interested in that. Where do I want the light to go? This is where I've kept a glass slide. Remember, you are all given glass slides in your second year, and if you break that glass slide in the exam, means you're going to fail the exam. So that glass slide, you know, where do you keep it? This place where you keep the slide. The slide is kept on a stage. So what is the name of that place? Slide is the hero. Hero is always going to be on the stage. So you keep the slide on the stage. Now tell me, do you want the light to go in all directions? Of course not. I want the light only to go to my slide. I want that light should go only to the slide. How do I make sure? Because here I have a tiny little thing below the stage. Can everyone see? Below the stage, we have something known as the condenser. Remember, there is something known as the condenser, and this condenser has an iris. Iris means diaphragm. So if you've ever noticed, when light goes into our eyes, what do we study? That light goes into the eyes and there's an iris which is going to regulate. Same way, this is a condenser. I can open and close it. If I open it, a lot of light will come. If I close it, light will stop coming. So basically, it's condensing. Light was going all here and there. Now what is it going to do? It's going to condense it. So that is what I call as a condenser. Now, having said that, the famous most question that you get is, condenser is present above the stage, below the stage, right of the stage, left of the stage. Simple. They won't give you a diagram. They'll just ask you theoretically. So where is a condenser present? Condenser is present below the stage. Very, very famous previous year question. Okay, having said that, let's move forward. So now all the light has been condensed onto the stage. What else do I see? You say that for some people, the stage, as per the eyesight, the stage has to be kept at an upper level. For some people, I will have to lower it because everyone has a different eyesight. So how am I going to move this stage up and down? If I want to move this stage up and down, I realize there are two knobs. Look at this very carefully. I'm showing you an unlabeled picture so that you can see the knobs. Can you see there is a big knob and there is a small knob? So if you want to move it majorly up and down, you use the big knob. If you want to move it, my vision and your vision, what must be clear to me, you'll say it's looking a little blurred to you. Few little focusing you want to do, then you use the small knob. So can I say in simple words that the big knob over here, that is going to be, or we'll label it here, the big knob over here is going to be the coarse adjustment, because that is telling you how it is going to do big movements up and down. And the fine one, the small one, is going to help in fine adjustments, right? So coming back, the big knob is going to be the coarse adjustment and the small one is going to be the fine adjustments. Okay, now finally you're looking at it. You're looking at the microscope. So what do you need to know? You are seeing that there are two lenses over here. This is where my eyes will be. This is where I am looking at it. So what lens do I want to call this? This is known as the eyepiece lens. This is referred to as the eyepiece. Okay. Having said that, you're looking at it, fine. But then I have a set of lenses here also. And what are these? These are said to be the objective lenses. So we have an eyepiece lens and we have an objective lens. Okay? But now comes the, because lens was the one that is going to zoom in and magnify everything for you. Hai na? So lens has to be the most important thing. Please remember that when I'm talking about the objective lens, I can see 1, 2, 3, 4. There are 4 different lenses involved out there. So let me get into a little bit more detail of that, right? Let me get into a little bit more detail and let me answer this question that in a light microscope, what are the two lenses? Objective and eyepiece. Eyepiece is the one that is obviously going to be where my eyes are. That gives me a magnification of 10 times. That gives me a magnification of 10 times. Whereas what does an objective lens do? Objective lenses, I told you that I could see 4 objective lenses. So let me write those 4 down. The smallest one is for scanning. The smallest one is only for scanning. Little bit of magnification it will give me, and that magnification is a 4 times magnification. Then I say I'm not happy with 4 times. Let me go to a little, bigger magnification. So first I go to low power and then I go to high power. Okay, low power and high power. So remember, low power is going to be 10x, and high power is going to be 40x. So how many lenses? 4, 10, 40. But finally I say I want to look at it under the oil and under maximum. What is oil immersion? Oil immersion is going to be 100x. So remember, 4, 10, 40, 100. These are the 4 lenses. Have a look at that picture again. Now you will visualize it over here. Could you see 4 lenses? So there are small, big, big, big. So 4, 10, 40, 100. That is how the lenses are. This is where it is. These are all the objective lenses. You can write 4, 10, 40, 100. And do you know how am I going? They are going to move around. They are going to revolve around. So as a result of which you will realize how are they revolving around. What is this known as? This is referred to as the neck piece. This is referred to as the neck piece because it's your neck. It's your neck which is roaming around like this. Na? So it's that entire round thing which is roaming around on what those 4 lenses are hanging. So you can move around and use whichever lens you want to. Having said that, yes, coming all the way back, now let's look at what is left that I had to teach you. You just don't have to learn this. At the end of the day, when you're looking into a microscope, everything that you've seen is getting added and coming onto you. So you are not bothered whether you are getting the magnification of objective or eyepiece. At the end of the day, you are going to see a magnified image. So if they ask you, what is going to be the total magnification? Very, very important point. Listen to me very, very carefully. If they ask you what is going to be the total magnification, you will first say, first tell me the magnification of these 4 objective lenses. First tell me the magnification of objective lenses because you know they are 4x, 10x, 40x, 100x. That you are going to multiply with whatever magnification you have on the eyepiece. But eyepiece lens was only one lens. Means every time it is going to be only 10x, 10x, 10x and 10x. So if I find out the total magnification, you just have to multiply them. So what is, if I'm using a scanner, what is the total magnification? 4 into 10, 40. If I'm using low power, what is it? 10 into 10, 100. If I'm using high power, means 40. 40 into 10, 400 times. And if I'm using 100x, 100 into 10, it is going to be 1000x. So if they will ask you what is, and this question is the most important, what is the maximum magnification that a light microscope can give you? The maximum magnification offered by a light microscope is 1000x. You have seen this question so many times in your previous years, haven't you? Okay, having said that, there's something extra that I want to teach, and that is a numerical aperture. Now, you don't have to go into too much of detail. I'll show you one picture. This much, see, I don't want you to learn these numbers. Please don't. Please don't. And when I genuinely say don't even try to for knowing extra knowledge because it's totally useless to know these numbers. I just want you to know that as your lenses were increasing from scanner to low to high to oil, I saw that the numerical aperture is also increasing. So I just want you to know what is numerical aperture. Have a look at this picture and life is sorted. So please remember, under this numerical aperture, I can see one angle forming. So this is a lens. Let me orient all of you. This is a lens. This is your slide or specimen and light is coming. Can you see numerical aperture is nothing but, numerical aperture is how much of light is getting see, light is not going to make a full angle. What is the lens making an angle with the object? So over here, if this is the object, I can see light is making this kind of an angle. If this is the object, I can see light is making this side kind of an angle. So remember, numerical aperture is n sine theta. Numerical aperture is n sine theta, where n is going to be the refractive index, which is usually around 1 or 1.5. And theta, you can see what is theta? Theta is half the angle. Theta is defined as half the angle. So numerical aperture is n sine theta. And what is n? N is the refractive index and theta is half the angle between the object and the lens. You just have to know that, look at these lenses. See, the lenses are from chota, bada, bada. There is a small lens, then there is a bigger lens, then there is an even bigger lens. And you will actually realize that as your lenses are increasing, the numerical aperture is increasing. So as the magnification increases, the numerical aperture increases. That is all that you have to know. Okay, having said that, coming all the way back, I think we are sorted when it comes to the light microscope. I want to do a very quick revision of all the parts so that there is no confusion. The first one that we had over here was obviously the light source, the light, transmitted light. Then what you have over here is the iris or diaphragm or condenser, whatever you may want to call it. The main hero is going to be on the stage. How do you move the stage up and down? Either you can move it by coarse adjustment or you can move it by fine adjustment. What is the lens system? The lens system, the one which I look inside, is the eyepiece. The other one is going to be the objective. How are you going to move the objective? We move it by the neck. So that is going to be the neck, or in some places it is also known as the nose piece, basically nose or neck. That is how they talk about it. So both, there are many books when mention nose or neck piece. Now, having said that, guys, I have explained everything to you for a light microscope from micro and path point of view. So going forward, your second year topic of microscopes is sorted. Okay, so having said that, having explained all of those, we can move on to little bit of variations. Now there is something known as a dark field microscope. So your mnemonic for light microscope had which kind of light, and dark field microscope has which kind of light? Listen carefully. Light microscope versus dark field. So remember, light microscope has transmitted light and dark field microscope is going to have reflected light. Something is going to reflect. That is why are you calling it dark? Because there's going to be a dark background, and the light that you are using is R, that is the dark light is going to be reflected light. So if you look very carefully, because I've got you the difference between a light microscope and a dark field microscope, between a light and a dark microscope. So do you notice what has happened over here? They have added what is extra. Basically, you'll want to know how is it different from a routine microscope. They have added something called a central aperture. So you will see that there is a hollow cone of light. The object is basically going to get illuminated because light is going to get reflected. So basically, this reflected light is going to illuminate your object. The object is going to be illuminated. Have a look at this picture. You'll never ever have any confusion. Can you see any slide? You will not even, agree, but this is actually the outline of the slide. But because our background and the slide, everything is dark out here. So I talk about the dark field microscope, but this was the slide, and that is going to be the black background. So first and foremost, why is there a dark background? Because which kind of light have I used? I have used the reflected light. And your next question is, under which scenario are you going to use it? This we are going to do again for the thin structures. Now, do you already know by now what are the thin structures? Like over here, all this spiral thing that is going on, this is actually the spirochetes. This is actually the spirochetes. But other than that, even for flagella, we can be using the dark field microscope. Okay, even for flagella, it's possible. I hope everyone's understood. Simple. Dark, everything's going to be dark and the object will be highlighted out. Why? Which light are you using? Reflected light. Very good. Now telling you something a little bit on the higher end. If you want to get a few extra marks, there is something known as an interference contrast microscope. Not something very usually asked, but something that you should know in case you are targeting extra edge exams. Okay, so first and foremost, this is for knowing internal nitty-gritty details. Basically, interference. When I want to interfere in every little thing, that is when I do an interference contrast microscope. So remember, it is going to reveal two things. What is inside the cell means the organelles, inside internal details, interference. And secondly, when you want to check out the constituents, how much of lipid is there, how much of proteins are there, how much of nucleic acid is there. So like I said, in an interference contrast microscope, you're genuinely interfering in every little detail of a cell. Now that could be what all organelles are present inside the cell, or how much of lipid is present, how much of protein is present, how much of nucleic acid is present, all that little interfering details. That is what interference contrast microscope helps you with. Moving on to the next microscope, which is examiner's favorite, and that is a fluorescent microscope. Now, first and foremost, mnemonic. As soon as you write fluorescent microscope, you will be asked what is the light that you use, and that is the ultraviolet light. So remember, for a fluorescent microscope, we use ultraviolet light. So let us basically revise all the lights. What was for a light microscope? Light with a T. So light was ending with a T. So we said for light microscope, it is transmitted. What is for dark field? Dark field was reflected light. And now I'm coming to fluorescent. Fluorescent is going to be ultraviolet light. Okay. Okay, so now, because if I show you this microscope, isn't it looking pretty much like any other light microscope? I mean, there's not much of a difference. You see the same light, you see the objective lenses, you see the eyepiece lenses. What is different? Something inside it. Some adapter, some too many wires are attached. Why? Because some rocket science is going on over here. So now when I look at the internal part, that is where I can see all the difference. So let me find out what is the inside part. So what do you think is this light source? I'll show you a more zoomed in picture. Have a look at this. What is this light source? This light source is the ultraviolet light. Right now, I just told you, you have to use an ultraviolet light. Okay, you have some filters. I can see ultraviolet light is going. But where have you kept your sample? What sample you've got from the patient? Here you have your specimen. Here you have your specimen. Now, what specimen will you put under fluorescent microscope? Now, I could use some, maybe it's a case of TB. If you're talking about microbiology, maybe it's a case of TB, or maybe it's a case of malaria. If you're talking about pathology, where do you use fluorescent microscope? Too much in pathology. You all know, guys, that fluorescent microscope is used a lot in kidney biopsies. याद है kidney में बहुत ज़्यादा use करते हैं. So let me talk about, say, kidney biopsy बात करते हैं, because now we are too bored of micro. Let us switch back to patho, right? Let us integrate. So for example, this was a kidney biopsy that was sent to you, and on a kidney biopsy, we have to do immunofluorescence, right? Okay, so now how will I do immunofluorescence? I'm just not going to put the kidney biopsy like that under the microscope. No, I am going to add some fluorescent dye. That is what I have to add. So you know which fluorescent dye will I add? That is going to depend on what sample do I have. So first, let me tell you the different types of fluorescent dyes. Everyone, listen to me very, very carefully. What are the different dyes? So if you say that I am suspecting a case of TB, the fluorescent dye that I will be using will either be auramine or it is going to be rhodamine. It is going to be auramine or it is going to be rhodamine. Okay. Next, now before I, because we are doing two subjects together, let's integrate in between subjects only. There is something called rhodamine, and I don't want all of you to get confused, because rhodamine was something that we were studying for copper. Okay, remember rhodamine, copper. But how do I differentiate? Because when I'm talking about rhodamine and rhodamine, there's only one alphabet difference. So remember that when they talk about MTB, M for Mycobacterium TB, which rhoda will you use? Rhodamine or rhodamine? I will use rhodamine for MTB, not rhodamine. Okay. Moving on. So next is MTB. After that, the next one that you have to know is malaria. So remember, for malaria, there's a very famous dye known as acridin orange. We use acridin orange. Moving on to the next one, when you're talking about a case of fungus. So I'm taking one bacteria, one parasite. Now it's time for a fungus. For fungus, we have CFW, calco flower white. And this is very easy to learn because I can correlate that the flower over here, flower is sounding like fluorescent. So I know calco flower white is flower for fungus, fluorescent for fungus, right? But this is all about micro. What is it about patho that I'm correlating? Do you all remember in pathology there was a technique called flow cytometry? What about flow cytometry should I be knowing? So flow cytometry was for all hematology samples when you were trying to find out CD4, CD8 and all of those, right? So remember, in flow cytometry, we use something known as FITC. That is fluoro isothiocyanate. If you can't learn the name, not a problem. At least learn this much, that FITC for flow cytometry. FITC for flow cytometry. Okay, all said and done. Now, the sample that I have over here, I said maybe it's a kidney sample. I can use any of these dyes. Maybe it's a TB wala sample. So now let me come back to microbiology. So if I say this is a TB wala sample, what stain will you add? Auramine, rhodamine. Fine. Now we realize that from the ultraviolet light, I had sent a light. But that light did not come till my eyes. That light is not coming. This is where my eyes are. Detector. This is where my eyes are. But the light, I realized, is not coming to my eyes. The light is getting deflected. Who is deflecting that light? And that is a dichroic mirror. So I see that there is a light going, and dichroic mirror has sent it first to the specimen. It has first gone and hit the target, the specimen. The specimen does some change so that the light which will be going back, see, you'll ask me, the light which is going back, how is that reaching your eyes? Why didn't the initial light reach your eyes? This specimen has done some magic. Initially, when the light was coming, it did not reach my eyes. It went to the specimen. But next time from the specimen, when this light came back, it came all the way to my eyes. You know why? Because initially the light that had come was of a shorter wavelength. And the specimen, lambda, I hope you all remember your basic physics, shorter wavelength, and the specimen has converted it into what? And that is the longer wavelength. So if someone asks you what is the principle of a fluorescent microscope, and that is the shorter wavelength is converted to, so when that light comes on a specimen, that specimen converts it into longer wavelength and sends it back. So now coming back, tell me all the questions very quickly. What is the light? Question number one, the light is fluorescent light. They will ask you that which is the microscope in which you use a dichroic mirror. That dichroic mirror is going to be used in the fancy fluorescent microscope. Question number 3, they will ask you what is the principle. So by now all of you know that a shorter wavelength is going to get converted into a longer wavelength. And finally they will ask you the names of the dyes, which you've already done, and this is one reference image in front of you. All of you can zoom in and see it's looking nice, fluorescent, shiny. I'll give you a background. This is actually a sputum specimen. This was a sputum sample. So these rods that you're seeing, these shiny rods that you're seeing, these are TB. These are Mycobacterium TB. So if this is Mycobacterium TB, what dye you must have used? The dye that I must have used was auramine and rhodamine, right? Okay, so now before I end this, there is also something that you have to know. Without adding any dye, I have not added any auramine, rhodamine, acridine, nothing. Something is shining on its own. So that is how you should be. You should have a light of your own. So these are the things which have a light of their own. And you don't need to add any colors. They will show you fluorescence on their own. So first, the Spora brothers. The Spora brothers are autofluorescents, means they are fluorescent even without adding any fluorescent dye to it. So what are the Spora brothers? We have Cyclospora and we have Isospora. So the Spora brothers are autofluorescent. Next, we have something in one in path. So we've done one microna. So now we'll do one path and one hemat. In pathology, remember, formalin is autofluorescent. Formalin, if you put formalin under UV light, it will start shining. Without any dye, it will shine. That is the reason, guys, I want to give you one correlation. Please remember, if the dermatologist calls you and says, I have done a skin biopsy of my patient and I want to send it for immunofluorescence. I want to send it for immunofluorescence. Can you please tell me that how should I send my biopsy? I'll say, please don't send it in formalin, because formalin to apna aap hi, forget the skin biopsy. Formalin is automatically going to start shining. I don't want that. Please send it in normal saline, not in formalin. So if the skin biopsy is being sent particularly for immunofluorescence, tell them that you will unnecessarily get a lot of immunofluorescence because formalin will keep shining on its own. That is one correlation. Second is another thing that is autofluorescent. Now going to hematology. NADPH is autofluorescent. So do you guys know there is a test in which NADPH or this concept is used? This concept is used in the fluorescent spot test for which anemia? Fluorescent spot test is done for which anemia, guys? It is done for G6PD deficiency. So in case you remember it, hats off. In case you don't, nothing to worry. Go to the hemat section. We've studied this under hematology in path. You know we've studied G6PD deficiency. I've told you there is a fluorescent spot test. And in that, what is fluorescent? That we use NADPH in our body. NADPH of our body is auto shine. You put it under UV light, it will shine. So basic concept is, if you put the Spora brothers under ultraviolet light, they will shine. If you put formalin under ultraviolet light, it will shine. If you put NADPH under ultraviolet light, it will shine. What was I saying again and again? Under which light? I hope you remember, it uses the ultraviolet light. One more dermatology application I want to tell you. In dermat, have you all studied wood lamp examination? Wood lamp examination, sometimes it shows you green fluorescence, yellow fluorescence. Yes. What is the light that you use? Why are you seeing a fluorescence? Or like skin mein kuch lesion hota hai and that is showing you fluorescence. Which light do you use? In wood lamp examination in dermatology, also we use the ultraviolet light. This is how you're supposed to study, guys. You have to integrate PAN 19 subjects, otherwise scattered concepts become very difficult. So in wood lamp, when I look at the skin under a wood lamp and the skin lesions are fluorescent, so if a fluorescence is seen, I've obviously used an ultraviolet light. Sorted. So fluorescent microscope is done. Let's move on to the next one. There is something called a phase contrast microscope. So first and foremost, if I look at it, it's looking like any other regular light microscope. I have a light at the bottom. There's a condenser. There is a stage. There is the, the objective lenses, then the eyes. Everything is looking the same. So how is it going to be different? I can see two things which are extra if you noticed. There is something called this annulus over here. Right, that is one thing. There is something called annulus or the annular diaphragm, and I can see there is some phase plate. There is a phase plate which is near the eyes. So basically I realize there is something added right at the bottom and there is something added right at the top near my eyes. So something added right at the bottom is known as the annular diaphragm. Something added right at the bottom is known as the annular diaphragm, whereas something that is added right at the top, that is going to be the annular phase plate, and these are creating differences in the phases. So you will obviously want to ask me, why are you creating any kind of difference in the phase at all? So this is done whenever there are different refractive indices. Whenever there are different refractive indices, that is when this problem or this microscope can be used. So why do you think there are different refractive indices? So for example, if this is a cell, inside the cell what all is there? There is a nucleus inside the cell, but apart from that there are so many other things. There's a ribosome, there is lysosome, there is endoplasmic reticulum, there is mitochondria, so many little different things. Everything will have a different refractive index. So all of them will have different refractive indices, and that is exactly what you have to convert and see that, okay, this has a different refractive index, this has a different refractive index, I have to see this also and this also. So whatever differences in the refractive index will be there, please remember, whatever differences in the refractive index will be there, that will show me different phases, and these different phases are then going to be converted into light amplitude. So this will result, obviously, if something is passing through a very high refractive index, light is passing through high refractive index, light is passing through low refractive index, the light is going to change. So that is going to result in change in the amplitude of light, and that is how the principle of this microscope goes ahead. This is going to result in change in amplitude of light. So I hope it will help you see everything, every microorganism in detail, all the internal details you can see, each and every ribosome, each and every mitochondria, all of those details will be seen. Okay, having said that, what is the next microscope that we have? The microscope that looks like a spaceship. And that microscope is electron microscope. Why do I say like a spaceship? Because you can very well see how it's become like an enclosed kind of an area, right? It's become like an extremely enclosed kind of an area. So why is the reason for that? I'll tell you. Firstly, what all do you know about it? Who was the scientist who discovered electron microscope? E for E, if you all remember, it was Ernst Ruska. Ernst Ruska identified electron microscope. And having said that, we further need to know, why is it looking like this spaceship? Because here, for the first time, I'm not using any light. In all the other cases, either I was using transmitted light, reflected light, UV light, some of the other light. Here I don't use light. Here I'm going to use a beam of electrons. And beam of electrons cannot travel like this out in the open. If you want electrons to travel, they'll be lost in the air. So you want it to be enclosed. Therefore, please remember, the medium is going to be vacuum. And that is the reason that this microscope is looking like so closed, as a spaceship type it is looking. So why is it looking? Because you want all the electrons to be enclosed in a particular place. Having said that, before I start off with different types of electron microscope, first I will have to tell you the basic differences between a light and an electron microscope. So let's start doing the differences first. I think the difference that you know is the resolution. Have we already done the resolutions earlier? Quick revision. When I'm talking about the resolution of the eye, it was only 0.2 mm. For light microscope, it was 0.2 microns, and for electron microscope we said 0.2 to 0.5 nanometers. So at least the resolution part is sorted for us. Baki sab let me have a look at. Okay, so first let me talk about the question that comes so many times, fixation. Fixation means if I send you a sample, let's go a little bit again. Whenever I feel that we are getting too monotonous in micro, I take you back to patho. I feel that way we'll correlate so many things. Okay, so I say that I want to do a kidney biopsy. There are two pieces of kidney biopsy that I've taken out. One piece I want to send towards light microscopy and one piece I want to send towards electron microscopy. So there are two pieces. One is this and there's another one for light, one for electron. So in which container will I send it? Will I just send that piece in regular water? No. I'll have to send it in a fluid. So for light microscope, what we use very commonly is 10% neutral buffered formalin. We use NBF. In simple words, we use 10% neutral buffered formalin. If it's written in the paper as NBF, don't get confused. Neutral buffered formalin. Formalin, the thing that irritates your eyes and makes your eyes water, that is formalin. Whereas for electron microscope, we are going to use 2 to 2.5% glutaraldehyde. We will be using 2 to 2.5% glutaraldehyde. So ultimately, I realize it's all about aldehydes. Because what is formalin? Formalin is also formaldehyde only, right? And over here I'm using glutaraldehyde. So basically I'm using aldehydes everywhere for fixation. For light microscope it is formalin. For electron microscope it is glutaraldehyde. I did a kidney biopsy. One part of the kidney biopsy I've sent for light microscope. One part of it I have sent for electron microscope. Now how are you going to look at it? First you will have to do a process known as embedding. Have a look at this, guys. Can you see this is the kidney biopsy over here and this is the kidney biopsy over here. But can you see how have you put them? What have you put them in? Over here, the biopsy has been put in paraffin wax. So when you embed it, when you put it inside something, I'm trying to embed all the concepts into your brain, right? So over here, this embedding has been done in wax. That is what you do for light microscope. But when you do the embedding in something very fancy, that is resin, electron microscope, so obviously it's expensive, na? It's going to use all expensive things. So when you embed it in resin, that is when you are dealing with an electron microscope. So simple. Where are you going to do the embedding? For light microscope, you are going to do it in paraffin wax, whereas in electron microscope, you are going to utilize resin. So now see, your samples are ready. What will you do? You'll say cut through it. Cut it. There's a blade. Cut it. Put it on a slide. Cut it, put it on a slide. Similarly, cut it, put it on a slide. I want to look at the slide under the microscope. So, jaldi se cut it, put it onto a slide. What slide are you talking about? Tell me, what slides do you get in your exams? What slides do you get? All of us get glass slides, and like I was saying, if you break a glass slide, you fail the exam. So, you know that for a light microscope, you use a glass slide. But electron microscope is not going to work like that. It needs everything expensive. For electron microscope, you need metallic slides, and usually these metal slides are made up of copper. Next, talking about the source. When I say source, you all know that in a light microscope, the source is going to be something different. I've already taught you what is the source. It's light. And which kind of light? It is going to be transmitted light. It is going to be transmitted light, whereas for electron microscope, there is no light, there is a source of electrons, and that will tell you the medium also. Because you are using a source of electrons, what medium do you maintain? You have to maintain that spaceship, you have to maintain the vacuum. Whereas for light microscope is you keep it here on the table, you keep it anywhere, it's out in the air, there's no problem, light can travel everywhere in the air. So the medium is going to be air. Having said that, we have wrapped up the most important table. This will help you again. Path, micro, both, everything fit in here. Coming back, this is the spaceship kind of an image that I was showing you. But nowadays, please remember, there are different types of electron microscopes. Now I'm talking solely about electron. There is something called SEM and TEM. SEM and TEM are going to be there. Now what is SEM and TEM? So let's go on to this. First and foremost, when I talk about SEM, scanning electron microscope, something is going to be scanning, and when I talk about transmission, it is going to be going through and through and through. So firstly, let me know, in both of them obviously I'm using electrons, but how are they different is what I need to know. So when I scan, now first before I start telling you all of this, get a basic outline with me. For example, you are reading a book. Okay, how do you read when you have your exam after 1 year? You go into every detail, every line, then you read, then you correlate from 2-3 sources, then you find errors, then you write to someone that, okay, this source has written this answer and that source has written that answer, because you have all the time in the world. You have your exam after 1.5 years, so you go into every little detail. And how much time does it take you? A lot of time. You spend approximately, you end up spending 2 weeks on every subject. So you take every little time possible, every little detail. Versus when your exam is after 2 days, how do you study then? Just flip through pages. Your speed becomes exponentially more, and you are just going scanning. Superficially, you're looking at, okay, main points, high yielding points, most important photos. You'll ask me for some 3-4 hour kind of rapid revision, right? That is exactly the difference between TEM and SEM. So when I'm talking about transmission electron microscope, please remember, I am going through every little detail. Every little detail I'm going to go through. So tell me, which type of study takes you more time? When you study every little detail or when you scan? Obviously, when I go into every little detail, I finish 1 page in 1 day. Can I say when I go into every little detail, I will have less sample viewed in the same time? When you go into every little detail, do you cover too much of syllabus? No, you cover 1 page per day. So you will have less sample. Whereas when you are superficially scanning, quickly flipping through pages, you will say, I will cover more syllabus. I will see more sample viewed in the same time. Okay. So what kind of electrons are you using? Simple process. When you are going into every, so see, your electrons will go through and through. If this hand of mine is the sample and this is your beam of electrons, your electrons have to go through and through. Then only you will see every little detail. So can I say you are using, in transmission, you are using transmitted electrons, whereas in scanning, when you just have to look at superficial, the electron will come, scatter. Electron will come, scatter. So hit, run, scattered electrons are going to be used. So simple. For scanning, scattered electrons. For transmission, transmitted electrons. Now talking about the dimension, remember, scanning is going to give you a 3-dimensional, because see, hit, run, the surface is being analyzed. The entire surface details are being analyzed. So this gives you a more 3D view. Transmission is more interested in what is happening inside, what is happening through and through. So this gives you, T2 is how you can learn. Transmission is going to give you a 2D view, whereas scanning is going to give you a 3D view. Finally, tell me who is going to tell you the internal details. By now you've understood who is going to tell you the internal details and who will tell you the surface details. So you know transmission is going through and through, so that is going to tell me the internal details. And what is scanning doing? Scanning is like hit and run, superficial. So who is going to tell me the surface details? And that is the scanning. So I think by now you have understood. You will always remember this, reading your course 1 year before the exam and 1 day before the exam. So 1 year before, you are like a transmission electron microscope. You are going into every little internal detail. You are studying 1 page. You are studying 1 page 1 day, very little details through and through. Transmission you are doing. Whereas now when you come to 1 day before the exam, jaldi, quick scanning through pages. You are using the touch upon it and leave it. Touch it, leave it, hit and run, scattered electrons. You are covering more amount of syllabus, more amount of samples, and you're looking at everything superficially, surface-wise. That is scanning electron. I hope with that mnemonic and that way of learning, you will always learn this, which makes us wrap up our microscopes, which also means that you're ready for some questions. So let's start. Question number 1. Scanning electron microscope is used to reveal what? Let's read. Is it the surface structures or the internal structures or both or none? So tell me, right now we have studied scanning. What are you doing in scanning? Superficial. So what is your answer? For scanning, I'm going to go through the surface structures. Okay. Next question. What is the maximum magnification strength that you get with a light microscope? Very important question. I told you, to get the magnification, you will have to multiply the objective and the eyepiece. So the maximum eyepiece is always going to be 10 times. Objective is going to be maximally 100 times. So what is the total magnification that I can get, which is maximum? That is the 100. 100 into 10, 1000x. Coming to the next one. In a light microscope, what function does a condenser serve? Does it increase the light intensity, focus the light on the sample, focus the light on your eyes, or reduce the glare? So do you remember that when I was showing you a microscope, if I come back to it, you will very well see that the condenser was focusing the light on the sample, right? This is where the condenser is present. So now if I make you answer that question, it's very obvious that it is going to focus the light onto the sample, not onto my eyes. So that's the answer over here. Let's take up the next question. A microscope that exposes specimens to ultraviolet light and forms an image with the emitted one at a different wavelength is called a dash microscope. So, firstly, as soon as I read ultraviolet light, I got my answer. Then they also said there's some change in wavelength, shorter to longer. I think everyone knows we are dealing with a fluorescent microscope over here. And do you remember the classical principle? The shorter wavelength was converted into a longer wavelength. Moving on to the next one. Which of the following structures are required in the microscope for taking this type of an image? Firstly, what type of an image is it? It's the classical shiny one, that is fluorescent. Now, if you remember, I told you what were the two things different in a fluorescent microscope from the others. Number one was the ultraviolet light. I don't see any light options out here. The other one was a mirror that we needed, and that was the dichroic mirror. Is that present in the options? Of course it is. Option C becomes your answer. Moving on to the last question that we have, after which we wrap up this segment. There's a microbiologist intern who wanted to study cells and organisms. His senior advised him to use a light microscope. What is the correct arrangement? Now, most of the students just learn, oh, okay, microscope and arrangement, but you forgot to read something essential. From the eye to the light source, means from eyes to light, from top to bottom they have asked you. So now tell me, none of the above to nahi hoga, obviously, let's rule that out. We will have to find that answer. So when I start from the eyes, which is the first lens that is going to come to my eyes? But I look into eyepiece. After that, what lens comes? Objective. And what is the bottommost? Condenser. Look at the other ones. In the other ones, they put eyepiece in the end. No, I'm starting off from the eye, so I have to have eyepiece in the first place, right? So eyepiece and then objective lens and then condenser becomes your answer. So the only trick over here was reading the question carefully, right? That brings us to the end of this segment, and we are done with the microscopes also. What do we start off with next is going to be the bacterial anatomy and the different shapes along with physiology that you have to study.